Hello everyone, my name is Ruiquin. Today our group is going to present this article published by drug maker Amgen about the results of KRAS inhibitor in human clinical trials. I will briefly introduce the background, then Alberto will explain the biochemistry, and Michael will then finish up with the results and discussion. The RAS gene family is the most frequently mutated oncogene in human cancers. Within this family, KRAS is the most prevalent variant and is particularly common in solid tumors. About 95% of pancreas cancers, 45% of colorectal cancers, and 35% of lung cancers are related to KRAS genes. G12C, the mutant Amgen scientists used, so named because the glycosyl at its 12th position is mutated to a cytosine. AMG 510 is a potent KRAS G12C covalent inhibitor with potential anti-neuroplastic activity. Although KRAS was one of the first oncogenes discovered, targeting its protein has been difficult due to the shape and the activation mechanisms. Only the inactive state of KRAS G12C that bonded to GDP is accessible to the inhibitor, allowing for accurate covalent blocking of oncogenetic signaling. Here's the signaling pathway of KRAS. On the left here, you can see this is the inactive form of KRAS that binds to GDP, and this is where the scientists were mainly focused on.
Hello, everyone. I will be touching upon the importance and the biochemical aspect of AMG 510 and its interaction with the mutant KRAS protein, KRAS G12C. Researchers vetted and screened 663 acrylamines for possible binding interaction with the pockets of inactive KRAS G12C protein structures. The reason they looked at the inactive form is because the active form of the mutant KRAS is relatively smooth and lacks any real pockets in its structure. The inactive form becomes more susceptible to opening up grooves which molecules can interact with. And what is AMG 510 and why is it important? Well, AMG 510 shown here in the bottom left is the first covalent inhibitor of KRAS G12C to make it into human clinical trials. And going back to the structure of the mutant KRAS protein, a mutation occurs at the 12th position where a glycine mutates to a cysteine, and this mutation is of particular interest because now the cysteine's sulfur is capable of making a covalent bond. AMG 510 does just that, and its methyl isopropyl substituted pyridine, this part of the molecule shown here in this location of the groove, is responsible for forming 25 van der Waals interactions within this point. And therefore, this molecule prevents the mutant KRAS G12C from becoming active and carrying out its function.
In summary, Amgen was able to discover AMG 510, which is a KRAS inhibitor. Although RAS was the first oncogene discovered, targeting its proteins has been difficult due to their shape and activation mechanisms. In this treatment, only the inactive state of KRAS G12C, that which is bound to GDP, is accessible to the inhibitors, allowing for accurate covalent bonding and blocking of oncogenic signaling. Amgen has found that AMG 510, a KRAS inhibitor, leads to regression of tumor growth for malignancies expressing the G12C mutation in a dose-dependent manner. And what this means is, in the top two graphs in the bottom left, the vehicle, which does not have any treatment, has the highest rates of tumor growth as compared to the various colored lines, which represent increasing amounts of AMG 510 treatment. And tumor growth decreases, as does the concentration of AMG 510. Furthermore, AMG 510 has been shown, when combined with cytotoxic or targeted agents, to result in enhanced efficacy. For example, AMG 510 in combination with carboplatin, a cytotoxic chemotherapy drug, as shown on the bottom right, has increased efficacy as compared to just carboplatin or AMG 510 alone. Furthermore, AMG 510 leads to increases in infiltration of T cells, which is augmented by immune checkpoint inhibitors, leading to a lasting cure of 90% of mice tested in the synergistic setting. So as can be seen in the top graph, the number of mice with circles, which lines with circles indicate tumor-free mice, is highest in the combination treatment. the green lines. Furthermore, we can see that the T cells are highest in AMG 510 or combination treatment on the bottom graph. Furthermore, in initial human clinical trials, when this was applied to humans, 54% achieved a partial response at one or more time points, and 46% achieved stable disease, for a disease control rate of 100%, meaning the treatment has been shown to be highly effective so far. In the future, it will be important to combine KRAS inhibitors with compounds that will encourage an inflammatory microenvironmental response, for example, upstream regulators or regulators of downstream bypass and feedback pathways. As can be seen in the heat map here, AMG 510 combined with cytotoxic or targeted agents results in increased and enhanced efficiency. Synergy scores for AMG 510 with targeted agents are represented by the heat map, with higher scores, darker red, denoting stronger synergy. Thus, the hope is that AMG 510 will be able to be applied in a personalized medicine approach to lead to even better patient outcomes and prognoses. These are our references, which we show the initial release, Science News releases, as well as the original article that the group published in cooperation with Amgen. We thank you for listening, and hope you have a great day. Bye.